Hi, my name's Brittany Lasseray, and today I'll be giving you a presentation on the biological field of epigenetics. We're going to look into what exactly is epigenetics and how is it related to potential disease causation. We're going to dive deeper into the three main tools that epigenetics works with, as well as how scientists began to focus more on epigenetics opposed to just looking at genes and the genome for disease causation. We're then going to dive deeper into certain diseases, lifestyle habits and thoughts and how they relate to epigenetics and are influenced by epigenetics. And finally, we are going to discuss potential future directions for this scientific field. So firstly, what is epigenetics? So epigenetics in the biological field looks at the study of how an organism's environment and thoughts can potentially influence the expressed final phenotype. It looks at how certain lifestyle habits and behaviors or environmental pressures can actually turn certain genes on and off. However, epigenetics highlights how these factors don't actually alter the genotype and the DNA itself, but alter the impact of gene expression. Through scientific research, scientists have discovered that there's three main tools that occur for epigenetic changes. The first one we're going to look at is DNA methylation. DNA methylation involves the chemical altering of the nucleotide base group cytosine, whereby it is chemically altered by either removing or adding a methyl group. This methylation can interrupt the normal transcription process, whereby proteins are blocked from being able to be attached to DNA and read. Therefore, the expressed gene might be a different one than initially intended. And this expressed gene may therefore arise in the phenotype. The second tool we're going to look at is histone modification. Now, histones are normally wrapped around DNA, but the way that they are wrapped around can alter the phenotype expressed. So in this structure, if the histones are packed too tightly together, the DNA can't actually be read, and therefore the gene won't be able to be produced, the protein, and therefore the gene is considered turned off. However, normally if the histone's structure is looser, the DNA can be read and the gene will therefore be turned on. The final tool that we're going to look at is non-coding RNA. So non-coding RNA's role is to actually attach and break down coding RNA after transcription has occurred, so that it's not actually used to make a protein and an abnormal protein. However, epigenetic alterations and selective potential environmental pressures can change this process, whereby this non-coding RNA isn't actually broken down and may actually be read in the transcription and translation process, which can therefore lead to abnormal proteins being developed and expressed in the phenotype. So what actually caused epigenetics research to become so popular? So scientists have always wondered what actually causes diseases and causes certain diseases to occur in some individuals over others. Now, the research into genes and their role in impacting the genotype and phenotype expressed in individuals has always been a topic of conversation. Through the previous scientists who have developed our understanding of genetics, it therefore, in the early 1990s, became a strong topic of direction in thought that diseases were directly gene-related. Therefore, scientists thought that if we were able to genome map the entire human genome, we would be able to cure every disease that occurs in the world. As that, in that point of time, they directly thought that genes and diseases were directly linked. Therefore, they decided to map the human genome and undertook projects including the Human Genome Project and ENCODE project. However, at the end of these projects, they realised that it wasn't actually specific genes that caused major diseases, as after they mapped the human genome, they weren't actually able to solve many diseases. So therefore, scientists began to wonder and speculate that there must actually be other factors that affect gene expression and disease causation. And this is where epigenetics came into the discussion. So through epigenetic studies, scientists have actually discovered that there is a strong relationship between epigenetic markers and cancer causation. They have discovered that the epigenetic tool DNA methylation has been directly linked to cancer in numerous studies. This research has dived deeper and discovered how the cytosine nucleotide base can become chemically altered and can cause major chromosome instability and alter different DNA sequencing processes such as protein synthesis. By this epigenetic marker being turned on through different lifestyle habits or factors, these newly altered genes can begin to uncontrollably produce oncogenes and oncomRNAs, which are mutated genes and markers that have the potential to cause cancer. This process of DNA methylation can also turn off the tumor suppressor genes in our bodies, such as miRNAs, thus leading to the individual being highly susceptible to the formation and phenotype expression of cancer. This connection between epigenetics causing cancer has led to many studies and actual potential breakthroughs in cancer treatments, including the introduction of epigenetic drugs to treat cancer. These drugs are being tested and trialled to be treating alongside other conventional treatment types of cancer, such as chemotherapy. So now we're going to look at epigenetic studies in relation to the lifestyle habit of smoking. So there's actually been several studies that have connected smoking with epigenetic markers and DNA methylation causation. So in a recent study of over 16,000 DNA samples, found out that people who smoked had 1/3 of their genes methylated differently to non-smoking individuals. This study therefore indicated that smoking can actually alter your gene expression and potentially impact and alter your expressed phenotype, thus potentially expressing different diseases. This study, however, actually also found that those who smoked and then stopped after 5 years, those 1/3 of the genes that were methylated differently had actually changed and reverted back to be similar to those test individuals who had never smoked before to start with. This actually gives a lot of hope for this lifestyle factor in these individuals, stating and potentially showing how these individuals, if they altered their lifestyle habits, can actually alter their genes and make them less susceptible to developing diseases based off epigenetic influence. Scientists have actually also looked at how epigenetics can be related to mental health. There has actually been several studies looking at how DNA methylation changes can actually occur in individuals who experience adverse mental health conditions in their life. Researchers at McGill University's Department of Pharmacology and Therapeutics studied mice in a scientific investigation and found that those mice who missed the nurturing and safety behaviours and habits of grooming and licking from their parents, which normally would occur, had altered genetic impacts in the long term. This study highlights how potentially individuals' thoughts and mental behaviours and health could also alter an individual's gene expression and phenotype in the future. However, this is still a relatively new area of study and much more research needs to dive deeper into this. So what is the future of epigenetics? So epigenetics has only recently come into full force as a field that scientists need to drastically focus on to help cure diseases. So therefore, it is still a relatively new developing field in terms of study and research. However, the next few years of research will focus heavily on epigenetics and focus on curing diseases in terms of the genes and epigenetic markers that it may display. Scientists and researchers might also focus on how altering our lifestyle habits can potentially actually make us more or less susceptible to certain diseases in the long run. Thank you.